Hello, my name is Dylan Friedman. I'm a rising sophomore at the University of Delaware, and this summer I had the incredible opportunity to work in Dr. Tanya Gressley's Animal Nutrition Lab as part of the UD Envision Undergraduate Research Program. Dr. Gressley's lab focuses on dairy nutrition through cow and silage experiments. Cows are ruminants, herbivorous mammals named for the largest and first chamber of their four-chambered stomach called the rumen. The rumen is responsible for microbial fermentation, allowing the breakdown and digestion of tough forage components in their feed, such as corn and alfalfa silage, which are types of preserved feeds. Milk production is an energy-intensive process for cows. Lysine is an essential amino acid that is critical for milk protein synthesis. For this reason, supplementing lysine to cows can optimize milk production and maintain good health after calving and throughout lactation, which lasts around 305 days for Holstein dairy cows. However, we can't just feed lysine to cows because lysine is very easily degraded by the rumen's microbes. Instead, lysine must be rumen protected, meaning that it has a coating that reduces rumen degradation but still allows for intestinal absorption. An effective product must have high bioavailability, which is measured as the percentage of administered lysine that gets absorbed by the body. The goal of my project was to test the bioavailability of different formulations of rumen-protected lysine products so that the company we're working with will potentially market the product that supplies the most bioavailable lysine. My experiment used 4 ruminally cannulated lactating Holstein dairy cows. Cows were moved to a tie-stall facility for a 7-day adaptation period before the start of the study. The experiment was conducted with each cow receiving each of 4 different rumen-protected lysine treatments over 4 experimental periods. We took blood samples from each cow during each period between 0 and 48 hours after administering the treatments. We immediately stored the samples on ice before bringing the samples back to the lab. The samples were then centrifuged to isolate plasma and pipetted into tubes to measure lysine concentration. We're waiting for the results, which will allow us to calculate the bioavailability of each treatment. The cows were milked daily at 5 a.m. and 3:30 p.m. At 8 a.m., we cleaned their stalls and placed down fresh bedding. Leftover feed was removed from feed bins, and new feed was weighed out in specific amounts for each cow. The feed was a TMR, or total mixed ration, made at the farm here and consisted of corn silage, alfalfa silage, and a grain mix. The amounts of each are specially calculated by our dairy nutritionist. After feeding and cleaning, we moved the cows back into their stalls. We started treatments at 9 a.m. and took blood from catheters every hour until 3 p.m., when samples were taken every 3 hours until 9 p.m. 24 and 48-hour samples were taken over the next 2 days. After each sampling, we sprayed the cows with ice-cold water to cool them off due to high temperatures during the study periods. We collected feed samples 3 times a week. Samples of TMR and corn and alfalfa silage were dried for 48 hours at 60 degrees to correct the ration for dry matter fluctuation. These samples will be sent out for wet chemical analysis for crude protein and other nutrient content. I had an amazing experience and thoroughly enjoyed the opportunity to not only conduct research as an undergraduate, but to have also gained significant large animal experience that will benefit me greatly as I continue on my path to becoming a veterinarian.